You may be 38, but your cells are 52. Your birthday is a date on a calendar. Your biological age is a biological fact, and the two don't have to agree. What's stranger still is that the gap between them, which can run to 10-15 years in either direction, is not fixed. It moves. And it moves in response to things you're already doing, or not doing, every single day. Here's what I want you to understand before we go any further. Your body has been keeping a record. Not in your memory, not in your habits, but inside the actual chemistry of your DNA. A running log written in molecules, updated continuously, and, as of fairly recently, readable. The science that makes this possible is called epigenetics, and the specific mechanism at the center of it is something called DNA methylation. That's the phrase you'll want to hold on to. Have you noticed that two people the same age can look and feel completely different? One sharp and energetic at 65, another already diminished at 55? Drop a comment, yes or no. And the answer to that question is exactly what this video is about. Before anything else, here's the simple version. Your DNA is like a master instruction manual, the same one in almost every cell in your body, but not every instruction gets read all the time. Your cells need a way to decide which pages are open and which ones are closed. DNA methylation is that system. Tiny chemical tags, methyl groups, each one a carbon atom with 3 hydrogens attached, get stuck onto specific sites along your DNA, and when they're there, they effectively silence that section of the instruction manual. When they're removed, that section opens again. Simple as a sticky note. Except the pattern of those sticky notes is, it turns out, a remarkably precise record of how old your biology actually is. Now here's what's underneath it. Your DNA contains roughly 28 million sites where methylation can occur, positions called CpG sites, where a cytosine base sits next to a guanine base along the strand. As you age, the pattern of methylation across these sites shifts in predictable ways. Some regions that were heavily methylated in youth gradually lose those tags. Others, which were largely unmethylated, accumulate them. The pattern doesn't drift randomly, it follows a trajectory. And that trajectory is consistent enough across human beings that researchers can now read it like a clock. Steve Horvath, a biostatistician at UCLA, published the first widely used version of this clock in 2013. He identified 353 specific CpG sites whose methylation status, taken together, could predict a person's age to within a median error of about 3 to 5 years, across multiple tissue types, from blood to brain to liver. The accuracy was surprising enough that many researchers initially assumed he'd made an error. He hadn't. What's since emerged is a family of these clocks, each calibrated slightly differently. Some, like Horvath's original, are good at predicting chronological age. Others, second-generation clocks like PhenoAge, developed by Morgan Levine and colleagues, are calibrated against biological outcomes, disease risk, functional decline, mortality. The distinction matters. A clock that tells you how old your DNA looks is useful. A clock that tells you how old your biology is behaving, how it's actually running, is something closer to a health forecast. In other words, one clock tells you the odometer reading. The other tells you how the engine's doing. The gap between those two numbers is what researchers call epigenetic age acceleration. And this is where it stops being abstract. People whose biological age runs 10 years ahead of their chronological age don't just have older-looking DNA. They have meaningfully higher rates of cardiovascular disease, cognitive decline, cancer. People whose biological age runs behind, whose cells read younger than their passport, live longer on average and spend more of that time in good health. The gap isn't decorative, it's predictive. I'll be honest, when I first spent serious time with this literature, what unsettled me wasn't the existence of the clock. It was how early the divergence starts. This isn't something that happens in your 60s. The trajectories begin separating in early adulthood. Two people at 30 can already have meaningfully different biological ages. Differences that compound quietly for decades before they surface as anything the person can feel. That's the short-term so what, and it lands harder than it sounds. The body you're running right now is not a neutral fact. It's a running consequence of everything that's happened to it. The question isn't what your biology will be, it's what it already is. Think of a methylation pattern like a library that's been annotated by everyone who ever used it. Every chapter that got dog-eared, every page that got highlighted, every section that was never opened, all of it leaves a trace. The library looks the same from outside. But open it, and you can read exactly how it was used and for how long. Your DNA is that library. Every physiological stress, every bout of chronic inflammation, every decade of inadequate sleep, it's all in there, annotated in methyl groups. Now, the The question that actually matters: can the annotations be changed? This is where the gap between what wellness culture says and what the research actually shows becomes important enough to slow down for. Here's the thing: the supplement industry got hold of this mechanism about 30 seconds after the research went public, and what followed was predictable: methylation support complexes, epigenetic reset protocols, things sold in amber bottles with serious-looking fonts. The actual evidence is considerably more specific, considerably less glamorous, and considerably more actionable than anything in those bottles. In 2021, a randomized controlled clinical trial published in the journal Aging, 43 healthy men aged 50 to 72, put participants through an 8-week program targeting DNA methylation directly. Diet, sleep, exercise, stress management, specific phytonutrients. No exotic interventions. The result: a statistically significant reduction in biological age of 3.23 years compared to controls. The same research group replicated a similar effect in women in a 2023 follow-up, with individual reductions ranging from just over 1 year to 11 years, averaging around 4.5. That upper end, 11 years and 8 weeks, requires a reluctant hedge. This was a small case series, not a large controlled trial, and the magnitude at the extreme end should be read as a signal, not a promise. The average effect, though, is consistent with the mechanistic picture. 11 years. I read that figure 3 times when I first encountered it, even discounting for the caveats. What the mechanism responds to is not random, it's specific, and the specificity is the point. Methylation reactions require methyl donors, folate, choline, betaine, cofactors that the body uses to physically manufacture and transfer those chemical tags. If the supply chain for methyl groups is compromised, the machinery that maintains the pattern runs less reliably. Dark leafy greens, eggs, legumes, liver, these aren't superfoods in any marketing sense. They're the raw materials for a specific biochemical process. The connection isn't metaphorical. Exercise does something distinct. Moderate aerobic work, roughly 30 minutes, 5 days a week, at 60 to 80% of maximum effort, is associated in multiple studies with lower epigenetic age acceleration. The mechanism isn't fully resolved, but the leading candidate involves inflammation. Chronic low-grade inflammation accelerates methylation aging, and sustained aerobic exercise is one of the most reliably anti-inflammatory things a body can do. Elite competitive athletes, worth noting, sometimes show the opposite pattern. Excess volume appears to impose its own epigenetic cost. The mechanism doesn't read your intention, it reads the load. Almost a quarter of the methylation sites used in the Horvath clock sit inside glucocorticoid response elements, regions of DNA that respond directly to cortisol, your primary stress hormone. Which is to say, chronic psychological stress isn't just bad for you in the vague sense that everyone already accepts and nobody fully acts on. It's writing itself into the methylation pattern. Directly, the breathing exercises prescribed in the Fitzgerald trial, twice daily, designed to activate the relaxation response, were not soft wellness filler. They were targeting a specific mechanism. 60 days of twice-daily practice has been shown in a separate study to measurably reduce epigenetic age on one of the methylation clocks. 20 minutes at a time. That's the dosing, such as it is. Sleep is the piece that gets the least specific coverage, possibly because it's harder to sell as a protocol. But the data on sleep deprivation and methylation aging is consistent. Insufficient sleep accelerates the clock. The threshold in the intervention literature is 7 hours. Not 7 as a round number, 7 because below it, the pattern of divergence between chronological and biological age reliably worsens. The medium-term picture across weeks and months of sustained change is now fairly well documented. The epigenetic clock moves. Not infinitely, not for everyone equally, and not in a way that's fully resolved mechanistically. The honest gap here is that we don't yet know how durable these changes are, how far they can be pushed, or whether they behave identically across different populations and age groups. The long-term data is still being collected. Some of what's been published is in small cohorts in specific age ranges, under controlled conditions that don't map cleanly onto ordinary life. The signal is real. The ceiling is unknown. What the mechanism tells you to do, if you take it seriously, is less like a protocol and more like a constraint. The system that regulates your methylation pattern runs on specific inputs: adequate methyl donors, sustained moderate movement, managed physiological stress, consistent sleep. The mechanism doesn't distinguish between someone who knows this and someone who doesn't. It runs on what it gets. Return to simplicity. Tiny chemical tags on your DNA are keeping score. The score changes. The inputs that change it are not secrets. They're just unglamorous. The peculiar thing about this particular mechanism, and I find this strange every time I think about it, is that it means the age on your cells is not the age on your license. It's closer to the age of the life you've been living inside that body. That's either reassuring or uncomfortable, depending on what that life has looked like. But it's not fixed, and that, honestly, is the point.